Well, so this presentation will be divided into 6 parts, and I will need to talk a little about the institute where I'm working on now, and uh, which I am now the director of operations there in Brazil. It's a very different institution there in Brazil. And then I'll talk a little about some projects or initiatives that we are conducting both by ourselves or supporting other groups in Brazil and together with the Ministry of Health and some national levels institutes there. And finally, I'll talk a little about the next steps we are planning. Well, the institute, before talking about the projects, I need to talk a little about the ITBS, is the acronym for All for Health Institute. And the institute emerged from an initiative from the largest bank there in Brazil in the very beginning of 2020. And we are a non-profit institution, and our budget came from this initiative, and we are funded in February 2021, and we are now working on this, trying to maintain networks, public health and surveillance, and also helping the Brazilian public health system to develop skills and facilitate uh, the public health to be ready for the next epidemics and pandemics. So we are a very tiny team. The core team is basically these 12 people, including our president, Dr. Maria Angela Simão, which is a former WHO director. I don't know if you know her, and myself as director of operations. We are currently hiring 2 other researchers, but we are basically researchers, specialists on data analysis, epidemiology, virology, and we have very talented team of support, including our journalists and our project manager and administrative support. Well, all of our projects or initiatives I will share with you are based on 3 foundational pillars: strengthening epidemiological surveillance networks, data analysis, and training and communication. So this is our institutional partners, our main maintainer, which has the name of the bank. It's a Brazilian bank, but it's a non-profit institution that is our maintainer. But we also have a public institution and some private institutions, including, I mean, Philippus, which is a public, pretty sure you may know, Philippus institution in Brazil. And well, we have currently a lot of projects or initiatives, both made by ourselves or with partners. But I will not talk about all of them, of course. It would take a long time for this. I've selected some of them that I think are most important for this audience. First one, just major, we call it HADIN. It's an acronym for Network of Data Acquiring and Analysis for Surveillance. And this initiative just emerged from this situation we have in Brazil, data blackout, the very beginning of the COVID-19 pandemic. The Ministry of Health was supposed to suffer a hacker attack or something like this, and they lost the capacity of gather data from the subnational levels and to provide information to decision makers. And at that time, we just began to think, think about different data sources for providing information on a timely manner to decisions on epidemiology and control methods at that time. So we just provide this mechanism, pipelines for acquiring data from different sources, mainly private sector laboratory tests, and to make all of this stuff here from the acquiring the data to providing a communication strategy in 3 or 4 days. I mean, in a timely manner, so it could be used for public health and epidemiological decisions. And this initiative was led by our uh, scientific coordinator, Dr. Anderson Brito, which is who is a virologist with great background on epidemiology as well. And these are some of our reports that were published uh, by using these pipelines. So here are some of these private sector laboratories, which we have some agreements on data sharing. So these laboratories share the data on tests, on diagnostic tests for all of pathogens. But at that time, we were most interested in respiratory ones. And all of this was possible because we had support from Dr. Steph. I don't know if you guys know her. She's a really experienced researcher there in Brazil. And she's also consulted from the ITPS Institute. But we also included public data as they were available for this time. But there's a huge challenge on putting this data together, collecting this data from different data sources. And this is being made since then. And we still keep this team on working on this data analysis pipelines, so we can provide this kind of data analysis. So here is probably the first report we provided. It was mostly focused on monitoring the Omicron variants emerging. So we could see here that through these reports, we could see exactly when the variant BA2 upsurged there in Brazil, and then all the variants from Omicron. And we showed for the first time to the public health institutions when BA1 was replaced by Delta variant there in Brazil. Remember that we had this blackout, so there were no information for this for public health things purposes. And then it just expanded this analysis for other respiratory pathogens, including flu and syncytial respiratory virus. And we stay analyzing COVID-19 as well. But we also expanded to arboviruses this year because I don't know if you know, but Brazil had this huge epidemic of dengue virus this year. We included dengue virus in these reports. And finally, the final outcome is not the reports or publication. We also move to informing the public health institution, and especially the traditional media and also social media and wherever, so we can inform these results to stakeholders, decision makers, and finally the general population. Well, but this is the first. It was mostly exclusively based on data. And now we are moving to include genome sequence to this project. So we are trying to select some of these samples so we can provide information on on genome sequence for these pathogens there in Brazil to public health. Thank you so much. Thank you so much. To provide information on genomic for public health purposes there in Brazil. So this is another project that is connected to the first one. So we are trying to make these arrangements, I mean, uh, agreements with these laboratories, so we can select some of these samples to make genome sequence. And we just began with dengue virus for obvious purpose. And this year, we just developed this pilot project on sequence genomics. So we provide some funding for some groups in Brazil, so they could sequence some samples of dengue virus, and we just allowed this data to be shared to the public in general. And we are very proud to say that almost 50% of the public available genomes of dengue virus in Brazil have some support of ITPS at that time. And we actually, you are pretty kind. And as you can see here, we are very concentrated in this region, southeast, but we're trying to expand to other regions, Brazil regions, and the next year. So it will require a share of funding or budget, and we're trying partnerships so we can expand both in scope and in scale. Something. Okay, thank you. So, but the project is not just to gather data on genome sequence. We also have these pipelines. Again, this process is headed by my colleague Vanderson Brito. He is involved with this initiative. I don't know if you guys know about it. This dengue lineage system. It's a pretty new proposal of. It's something like pangolin system. It's a proposal of naming lineages for dengue. The idea is to provide some background on naming these lineages of dengue virus, so you avoid the things of dengue cosmopolitan lineage, or I don't know, Brazilian lineage or something like this. And what they provide here, what they propose here is this naming not working, but I need to point here. This naming. Probably I can make it old school manner. So the names are supposed to have this pattern where the first number refers to the serotype. So this I don't know, these red colors, which are majority of the cases here, for example, are 1, 5 in Roman numbers, and E. So the 1 here represents the serotype 1. So dengue virus serotype 1, lineage 5, and sublineage E. That's the idea. So this, sorry. Yeah. So this blue one, for example, here is serotype 2, lineage 3, and sublineage C 1.1. So that's the idea, is to provide some pattern to naming this lineage from dengue, and this system is available. If you can scan this QR code, you can check how the system works. It's a pretty new initiative, so And the idea is to get this data from genome sequence and to put into this system through a pipeline so we can provide this, so we can provide some pattern naming this identifiers unit. We are working on this by this time, so we just included this analysis to our reports um, that I will share soon. Thank you. So if you can see all the details on the analysis of this genome sequence analysis, you can scan this QR code and check more details about this, about all this here. Okay, so you can check all the details on the phylogenetic analysis that were made by our team, and both for serotype 1 and serotype 2 dengue virus. And all of this is available through these reports we share at our site, and you can check by scanning this QR code as well. What is it? Artist? Probably you can make real-time translation by using some tool, the translation tool. Yeah, I mean, I think the you can do it by using Google Translate, for example. I hope so. So this next initiative I like the most because not only because it's out in the deepest Amazon, and I am from the Amazon, but also because it's a little bit disrupting, considering the Brazilian surveillance protocols in general. So the idea here is that these indigenous people, the riverines and people that lives in this region, has this pretty close relationship to wild animals, both by hunting or by living with them like pets. And I'm talking about non-human primates and birds and whatever. Anything that lives can be both hunting or pets as well. So uh, these indigenous populations that live in this region, especially in this region, there is a border between Brazil, Colombia, and Peru, a place called Tabatinga, which is pretty nice place. It's a white pig and so. Um, this population, wild wild pig. So these indigenous populations are pretty exposed to all of sort of pathogens there, but we have no idea which part of the pathogens are exactly interacting with this population. So they used to hunt and to have culture practices and to live pretty close with these animals and of course with the pathogens that live inside these animals. So Brazil has the most biodiversity in the world, and most of them we do not exactly know about something about them. We have about 700 new animals species discovered in an annual basis. And and you know that all of these animals have these pathogens, and we have no idea of the biodiversity uh, of pathogens that lives with these animals. And there is a clear intersection of infections and emerging disease and social vulnerability there in this region. I'm talking again about the deepest Amazon. It's pretty difficult to implement policies on public health in this kind of place. And there we have probably the most at-risk populations for infections and emerging disease living in these pretty distant places. So these special populations are pretty neglected by the official health system, and they probably face a lot of problems with logistics and services offered and whatever. And they have a very complicated health system there because you have all of these levels of health service being uh, available for them. But the first one is what we call primary health unit, which has very limited service being offered to the population. And I would say that the official surveillance could be available in the third level only, which is pretty distant from the original place we live in. And what we are planning is to move these protocols first level by empowering population with knowledge about surveillance and pathogens circulating in their uh, location. So the project by itself is quite simple. We want to sampling the animals that are critical to the population, but also sampling the population itself and the environment, and to browse all of this information to look for uh, risks in a surveillance, risk for the surveillance, I mean, risk for the health of the population actually. And well, this is a community-based zoonotic surveillance project that wants to measure the risk uh, uh, of the population itself and to provide some uh, based information to this community so we can have system itself working correctly there in these settlements. So the project itself, it's, as I said, community engagement, data science and biobanks, survey and to promote health system preparedness for emerging disease in remote areas. It's classical One Health approach project there in the Brazilian Amazon. And we have three main objectives. The first one is to assess exposure to new emerging diseases with potential zoonotic human transmission, to leverage community-based surveillance, biobank, and secondary data to develop prediction and monitoring systems for timely surveillance. And finally, to translate community surveillance and GUAI models, that is this spin-off on the project, into health system preparedness too, so we can planning health campaigns to these special populations. Well, and finally, this last project we are conducting together with, it was actually proposed by Dr. Morel, which most of you may know, and Dr. Gabriel Walla, which is our consultant there in the ITPS, who is a virologist and works for Fiocruz as well. So the idea here is to provide this database and biobank of biodiversity, of pathogens biodiversity in Brazil. It's supposed to be a network that can provide information for all of Brazilian biomes. And we already have these samples for sequencing. And of course, the ITPS will provide some funding for this project as well. So the idea is to sequence But that's okay. Sequence is more than 8,000 samples from non-human primates, birds, mosquitoes, vectors in general, and baits and rodents, wherever, from the north to the south of Brazil, so we can have an idea of what's circulating there in terms of pathogens. So the project, it's focused on having standard procedures, making this, of course, uh, biosecurity information as well, and but mostly providing a database on genomic biodiversity and a biobank as well to research and development. And finally, the next steps, I'm just finishing the presentation. So the idea is to prove the value of these tools to the public health surveillance and search for partnerships so we can expand both in scope and coverage of these projects and transfer technology and knowledge to the Ministry of Health and national instances. And finally, to help the SUS, which is the public health system in Brazil, be ready for the next academic pandemic. Thank you for the attention. Thank you for the invitation.
Okay, thank you for the presentation. Really nice talk, especially about the bio project. I remember several years ago, we wanted to initiate this global viral project. But I think there's some difficulty to coordinate different countries. I think Montreal reached the beginning with the regional viral project first, then maybe perhaps the global viral. So any questions from the audience? Okay, so start with me, okay? Yeah. So because for the viral project, I think we need a lot of sequencing machine and also the reagents. So I'm just wondering how it is established in Brazil, Amazon regions now.
Well, I'm probably not the best person to answer this question, but I know that Morel has access to these facilities and sequence machines and whatever. So I think the problem is maybe reagents and wherever, but machines are not a problem exactly. But in Brazil, we have this policy. I think the public health laboratories have sequence equipments, so they could eventually make all this sequence project. But I think this is uh, a research initiative, not exactly public. So maybe this project will be held in the Cyprus institution, probably.
Will you say something? No, I never disagree with you. I just want to compliment. So, in Brazil, the Ministry of Health has sponsored what they call Genome SUS, which is the public health system. And this initiative started out, they completed, they already have sequenced 20,000 genomes. The second wave now is another 20,000. And if the ministry keeps financing, the idea is to have 100,000 human genomes sequenced in the next 4 years. In terms of sequencing facilities, we do have, you know, machines like, Vanderson said, to sequence over 100,000 genomes a year already installed. A lot of them Illumina, but in some places like Fiocruz, there is a T7. And also private now DASA, which was one of
of the private labs that Vanderson mentioned has switched also to MGI. So there are many places now that are looking into PGI and GI sequencers, but Illumina still has like for this human genome project. We have 3X plus, and then a bunch of 6,000 that are involved. This, but so I guess that of course Fiocruz has a lot of the smaller MGI sequencers for the viral projects. And in fact, Marco Krieger, at the Vice Presidency of Production and Innovation, the T7, that's Fiocruz, they just had an RFA for projects, and then they contemplated the research groups there. They're going to be sequencing 3,000 human genomes and 2,000 viral genomes using the T7. So I answered.
Something. Yeah. So the second question is, I noticed the operator bias is now present in Brazil, also the usage and operator bias. So I think it's mosquito borne, not anti-borne or mosquito borne.
It's not exactly a mosquito, but we can call it
Okay,
mosquito
okay,
now.
yeah. So how about the situations now in Brazil about this virus practice?
Well, it's not exactly new to us in Brazil, because it's a virus that circulates in the Amazon region long time ago. And the new situation is that the virus is spread all around Brazil and other countries in the Americas, mostly in the Amazon, but not restricted to that region now. And we will probably face a new epidemic in the end of this year and probably the beginning of the new year. And we are expecting for the worst. Virus had this mutation, probably in the Amazon region, in the years ago maybe. And this new variant is somehow more, say, not exactly. Christmas. Yes. Maybe Patricia can talk a little about. So, because I'm not.
Yeah, do we have vaccines or just now?
Well, vaccine.
And no vaccine.
And it's quite complicated to control the mosquito as well, because it lives in the. So we cannot use insecticides or something like this. So what to do? It's a. Just forgot this species. Yeah. Paraná, Santa Catarina. Not vaccinated. It is. Yeah, including the Atlantic forest. So the vector is everywhere, and now the virus is everywhere. Well, it's a huge public health problem there. Face. I cannot think about a solution easily waiting to solve it.
We can do something together, right? Okay, so any questions? Okay, so thanks again. So as a final step, I think we will give some certificate for all of the three speakers. So I invite Professor Wang together to give the certificate to our audible speakers. Okay, first of all, then let's invite the boss up. So thanks very much. And also Antonio, yeah. Antonio. So last, okay, Vanderson, yeah. Thanks for your contributions. Okay, so lastly, we will have a picture together. Okay. Get your first name. And also Andrew, please come. Andrew, you are the fourth delegate. Thank you. 一起啊，连水你也一起来。Yeah. So okay, so I think I want to add this could be things. So thanks again for your questions. And we hope this is that we can have more collaborations in the future. Definitely. Tropical medicine or tropical disease, I think it will affect all of them. Right? Thank you very much.